for the Exploring Our Oceans MOOC. I'd like to introduce a different team presenting today. So my name is Rachel Mills and I'm the Head of Department of Ocean and Earth Science, and you met me last week. But to my right here, I've got John Copley, who you'll recognise as one of the lead educators from the Exploring Our Oceans MOOC. So John's obviously a marine biologist, and he's going to be answering questions that have come up on the forum about biology. To John's right, we've got Emma Cavan. Emma, you'll have met on the forum. She's been answering a lot of questions, and again, mainly about marine biology, but she's also, because Emma did an undergraduate degree with us, she's got a lot of understanding of all of the subjects in oceanography. And then to Emma's right, we've got Rui Vieira. I pronounced that right?
Yes.
And Rui, again, is a PhD student who, again, is a marine biologist. So I'm a chemist, as you know from this week, and between us, we're going to try and answer any of the remaining questions that have come up over the last few days. So let's start with week 3. Week 3 was about marine biology, and I've got 3 biologists here, and we're going to sort of try and answer some of your more interesting questions and then make some general comments, and then we'll move on to chemistry in about 20 minutes time. So let's start with a question. What would you like to start with?
Why don't we start with the siphonophores?
Okay, so we've got a question here from Dorothy Grimshaw. Dorothy says, how do they know that the siphonophores are a colony of cloned animals rather than just one animal? So who would like to start with that?
Fine. So we know they're a colony of cloned animals because every one of the polyps has the same genes, it has the same genome. So we know that they're all clones of the same original individual, or if you think it was one individual, if you like. So how do the different polyps, because we have polyps for feeding, we have polyps for reproduction, they have polyps specialized for different roles, they all have exactly the same genetic blueprint, and that's one of the wonders of developmental biology, how you have the same blueprint, but you can actually encode for different body forms, different functions by expressing, turning on and off different genes during development. And then the whole thing is so wonderfully somehow coordinated together. So functionally, the colony we can think of as one organism, and genetically it's one organism, and yet within it we have this differentiation of function amongst its the members of the colony.
That's great. Anyone else want to comment on that? Did anyone work on siphonophores? Okay, well, let's move on down my list of biology questions, and let's talk a little bit about, let's go to this question here from Jane Smith. Okay, so Jane asks us, are there a lot of labs worldwide who are analysing new species? Is there often some overlap or some new species being discovered at the same time? And I guess this is for John, really, who's been leading on this.
Well, it can happen, but it's quite rare. So, for example, if, let's take some of the creatures you've been looking at from the Cayman Trough deep sea vents. Obviously, when we went there and collected specimens for the first time, we had the only specimens. So we then know who's working on describing the new species from those. If you have a site that several countries were visiting in the same time period and collecting material, then we'd be talking to each other to get the most out of our projects anyway. For example, if we've been there first and our American colleagues then went there after us, we can help them because we've got the map from having been there first. And we would then talk about, okay, we're working on describing these. You might be sampling other animals that we've seen, you might want to describe those, or we'll share the work up between ourselves. In the sort of rare event, and it tends to be back in history a little bit more when there were more unknown species on land and so on, yes, you could in theory have different people analysing them around the world, and in that case, it's whoever publishes first gets the sort of official name. And then other names, if they come out unbeknownst, and it's much rarer today with modern communication, but if that did happen, they would be a synonym of and recognised as actually the same as, and it might be called this by other people, but this is the official name. So there's a sort of order of precedence.
Okay, that's good. Okay, I'm going to move on to a question here from Rui, and I think the first question is from Peter Harding. So Peter asks, I'm just wondering at the evolutionary rate of change for deep water fish. Some of them look like living fossils. And of course, the coelacanth was indeed a living fossil. Is evolutionary change slower in the deep ocean?
Yeah, this was actually one of the good questions of this week, particularly because I work with deep water fish. And a good example of living fossils are the chimeras. And so we know that in deep sea, low temperatures and high pressure, they have great influence on the evolutionary rates. So these deep water fish they tend to have low metabolic rates, and these low metabolic rates will slow down the evolution of these deepwater fish. That's why at the shallow depth, where we have more, let's say, chaotic environment, the evolutionary rates are higher than in the deep sea. So we have a much more diversity of, for example, deepwater fishes or any other animals than in the deep sea.
So is this what you're studying for your PhD?
It's part of my PhD, yes. I'm part of my project will give a contribution on the understanding of the coexistence of deepwater fish at in the deep sea, and will give some insights on the metabolic rates in the deep sea.
Okay, so John, do you want to make any comments? I mean, John's obviously worked a lot on evolution from a different perspective, looking at biogeography of the deep ocean.
Yes, so it's very interesting, and you know, that we see this pattern in the deep water fish. There are a few deep sea environments where there is more rapid environmental change and higher metabolic rates, but those really are the exceptions. So the deep sea vents are perhaps a little bit different to the non-vents of that deep sea environment. So you know, there are some interesting comparisons yet to be made, I think, in looking at this question.
Well, I think the next question really follows on from that, Rui. So the next question comes from Tim Thornton, and he asks, why does high pressure reduce the metabolic rate? Is it because the concentration of dissolved oxygen in the water is lower? If there is no gas in the animal, then compression shouldn't be an issue, and chemical reactions normally run faster under higher pressure.
So again, I will give the example of deepwater fish, because of course it's my case studies. The metabolic theory is still under very big discussion. Some scientists, they propose that the temperature is ruling the metabolic theory or the metabolic rates. Others say that is the pressure. Others say that is the visual interactions. Basically, no one really knows, and it seems that different theories can explain different patterns. For example, the very deepwater fish, it seems that is the pressure that is ruling these metabolic rates. Why? Because the enzymes, these deepwater fish, they require some enzymes to keep their body stable. It's called, for example, one exam is the TMAO that keeps the hydrostatic pressure continue. If the animal, they don't have these enzymes, the body will collapse. So with the water pressure. So it really it seems that for some animals we can apply some theories, for some others we cannot apply the same theory, and there is a huge discussion on what is theory that better explains these metabolic rates, the coexistence of the deep living organisms, and of course the evolution on the deep sea.
John, do you want to comment on that? Especially the bit about gas and about gas inside bodies of deep
Yes.
sea organisms.
So okay, most deep sea animals don't have gas inside them, so that Tim is quite right. That isn't a factor. But as Rui said, one of the issues is enzyme structure and function under high pressure. It's actually about water getting trapped rather than gas. And so having these molecules like TMAO pull the water off the unfolded protein so that they do their jobs properly is key. And again, one of the interesting things about the deep sea is we have actually this variety of environments. So yes, separating temperature and pressure has always been difficult in the past because as we go deeper, the pressure increases and generally it gets cold too. So is it cold temperature? Is it high pressure? Of course, the place to go and test this are deep sea vents where we have high pressure because we're deep and where the animals are living, yeah, you know, maybe 20, 25 degrees. So we have similar to surface water, shallow water surface shallow water temperature. So TMAO, this wonder chemical that helps bony fish, and actually the shark-like fish as well, and the decapod crustaceans cope with high pressure. Yes, as you go deeper, you get more and more of it, and that may actually limit the depth to which some of these groups of animals can reach, because eventually you get other problems from having so much of it. New results that we haven't yet published. So pinch of salt, hasn't been through peer review, and we always have to be careful in case we've made a mistake somewhere. But I had a undergraduate project student last year doing some analysis on this, on some samples from vents. I've had students in previous years, and we've now pulled all the data together. And what's really exciting is deep sea vents, where we have the high pressure, but we have elevated temperature compared to non-vent deep sea. We find about a third the level of TMAO that we would expect for the depth. So it suggests that actually temperature, rather than pressure alone, is pretty key. But we have to go to these contrasting environments to kind of get nature to do the experiment for us. So we'll be working that up and publishing it. And in fact, we had a meeting about it this week. So hopefully in a few months' time, we should have an interesting paper to add to that debate.
So far, what I understood about these processes is that at different depth, even if the species are similar, there are different factors that controls the
and body size is another big one.
Yes.
So metabolic rate tends to scale with body size, and that will affect the amount of TMAO as well.
Yep.
So yes.
And I think that's great that undergraduates that are studying here are doing research in your lab and then writing papers. I mean, that will get you fantastic jobs. While we've got Rui here, who is one of our PhD students,
yes.
I want to know how you got here. You're obviously from Portugal.
Yes.
So tell us your story about how you got here to be studying at Southampton at the National Oceanography Centre.
So to tell why I came here, I have to start with my undergrad. Yeah. So when I finished my undergrad, I moved to the Azores Islands in the middle of the Atlantic, and I was doing an internship there with something totally not related with marine science. I was working with maize and apples. And
so you did a biology undergraduate?
I did marine biology and biotechnology, and I did my internship in biotechnology. But it was in the Azores that I get more contact with the deep sea and with working with the fishes, and I got very interested. When I finished my my internship, I moved to the Algarve in the south of Portugal to study marine ecology, and I did my master thesis under the scope of international program about the Antarctic midwater environment, where I studied midwater fish. And this was my really first contact with the deep sea. And after, while doing my master's, I got invited to join a research cruise in Cape Verde, where we study some seamounts around there. And yeah, it was the beginning of my deep water career. And then I moved to Aveiro, where I keep working with the deep water environments. And actually, Southampton was always my very first choice to do the PhD. And here I am working with the deep water fish in Southampton.
And obviously, we have a lot of students from all parts of the world. And so our graduate school has maybe 200 students in it, and they come from a real wide diversity of places.
Yeah.
So Emma here is also studying for a PhD. You've been in Southampton for how long?
I think this is going into my 7th year now.
Right. So she obviously really likes it here in Southampton. Again, while we're on the subject, tell us your story of what you did at undergraduate, how you got there, and what you're doing now.
So I did marine biology at the university and the Oceanography Centre here. I chose to do the 4-year programme because it included a 4th year master's year and a trip to Bermuda, so that was quite a strong selling point. The Southampton and the National Oceanography Centre was my first choice in the UK for universities at the age of 18, mostly just because of its how famous it is, and yeah, it's supposed to be the best in the UK for oceanographic research. Certainly one of the biggest. So yeah, so it was my first choice, got accepted, and I think in my 3rd or 4th year I knew that I wanted to do a PhD and to stay in academia and research. I didn't even really look anywhere else. So I don't know what they do to you here, but they just put something in the water to make you stay. Yeah, and so I applied for a PhD. I actually applied for one in much more ecology based than what I do now. So I applied for one looking at using isotopes to map fish distribution with Clive's now supervisor. But they wouldn't fund it. And then I very randomly got emailed by someone else who works here, who's more on the biological oceanography side, so plankton and all the smaller life in the ocean, saying that they were going to offer me 2 different PhDs that I could choose from. One was to do with ocean fertilization. I actually chose not to specify in that because I'm not really sure what I think about that as an actual solution. So yeah, that was quite popular in the online this week. And so now I'm studying basically sinking plankton poo, which is a huge drawdown of atmospheric carbon into the deep ocean and feeds a lot of the life that Rui and John look at. So I do much more surface orientated stuff and with smaller animals, but I get to go to sea a lot, and then I see all the big animals like the whales and penguins and stuff. So that's cool.
And just to go back to, what did you study at school or sixth in college to get yourself
I did
into marine biology?
Maths, biology and chemistry, and I also did philosophy and ethics at AS level, because most universities you only need 3 scientific A levels. Is that still correct?
Yeah.
Yeah, so I thought it was really important to do something just a bit different and interesting, and actually it really helped my writing to do philosophy and ethics, because they realised I was scientific and that my English was really poor, so they kind of boosted me up. But yeah, maths, biology and chemistry is what I did, and that was very useful to do. Yeah, very useful to have that broad knowledge.
Okay, let's move on a bit then, and because I think we're going to go for about 45 minutes today, because I know a lot of you got other things to do. Let's move a little bit on to week 4 and get on to this question about fertilizing the oceans with iron as a solution for drawing carbon dioxide out of the atmosphere. But before we do that, I just wanted to say a few things about week 4. So first of all, I know a lot of you are really struggling with the maths. Good on you, those of you that are doing it, but as a lot of the mentors have said online, it doesn't matter if you don't want to do the maths. You can learn all the information from the course without the maths. The reason why we put all that material in there is people learn differently. Some people learn by doing calculations and understanding the magnitude of things. Some people will look at an infographic and look at it pictorially. Some people will read the text and learn by doing that. So I think what we're trying to do is provide different ways of seeing the problem. So well done those of you that have done it. And I wanted to start by talking about making salt. So I think there's been quite a lot of debate about actually how long people have been extracting salt from the oceans and how they do it. And I made this comment in the video, I think, about the Romans learning how to make salt taste different by using different fractions of it. So I thought we'd start by just talking about that. Clearly, the easiest way to make salt out of seawater is to evaporate the water and collect the residue. And to be honest, we've been doing that forever. And we still do it. So the French still do it. In fact, the Portuguese still do it.
Yeah,
exactly.
Yeah.
And that residue is relatively moist. It's sort of grey in colour. Yes. It's not white. No. But it is a delicacy. And it tastes very distinctively of the local region where it's been evaporated. And everything that was in that salt, in that water, will be in the residue that is bottled up and sold at exorbitant expense. And it tends to be moist because the crystals are what we call hygroscopic. They don't dry out well, so it doesn't work well in a salt shaker. It doesn't work well in a grinder. You have to spoon it in, but it is a great delicacy. But most of us prefer our salt to be more refined, and even the Romans wanted this. Most of us want to actually exclude maybe some of the other minerals that are in there that give it a slightly weird taste. So, as I pointed out in the video, every element in the solar system on the planet is present in the ocean, and obviously sodium and chloride are the dominant ones, but there's a lot of calcium, there's a lot of magnesium, there's a lot of potassium, there's a lot of sulfate, and there's a lot of carbonate from the carbon dioxide. So a lot of these other components of seawater are making minerals when you dry out the seawater. Calcium carbonate, chalk, tastes really chalky. Potassium chloride doesn't taste great either. Magnesium sulfate is really, really bitter. Epsom salts, it doesn't taste good at all. So these other salts within the sea salt matrix make it taste either bitter or chalky. And we can exclude these by either skimming off the surface and getting rid of the surface scum, or leaving behind the last dregs, which will have the magnesium sulfates in. So what the Romans learned to do was learn how to rake out the crystals they wanted from the salt pans on the beach where everything was evaporating. And it's the sodium chloride salts that is the more refined white crystals that we're more familiar with. But I think it's really interesting that over time, people have actually, I mean, some people still use the moist residue of everything that's in there, and it's a delicacy.
In Portugal, we're still doing the very traditional way.
Mm. And is it very expensive?
No, but now there's crystals, this salt is a kind of gourmet.
Yeah.
So yeah, we export this gourmet salt to China, to France at a very high prices.
Yeah. Okay, so I was going to talk about two other things, I think. Let's talk about fertilizing the oceans with iron, because I think that's something that a lot of you have commented on the forum. It's a sort of, it comes out of our discussion about how iron is limited in the ocean in many areas. There's such low levels of iron dissolved in seawater that there isn't enough for plankton to thrive. And the main areas where this iron limitation occurs are in the Southern Ocean around Antarctica, in the equatorial Pacific, and in the sort of Northeast Pacific. But in fact, the more we look, the more research we do, we find that a lot of the ocean is either seasonally, so at certain times of year, iron limited, or iron limited in ways we just didn't understand because it was masked by other things. So in fact, iron limitation is virtually everywhere we go, we find some manifestation of it. So for the last couple of decades, certainly since the mid-90s, there have been scientific expeditions to seed the oceans with iron. And these have been variably successful, and some of them incredibly successful, some of them have been rather disastrous. But essentially the idea is you take iron sulfate, which is very soluble, and you essentially disperse it from the ship in a controlled manner, and then you monitor what happens. You monitor how the plankton utilize that iron, you monitor them from space using satellites because you can see the chlorophyll, and you look at how it's dispersed, and then you look at what happens to it. Does it sink down into the ocean? So I'm going to pass over to Emma to comment on your views on, is it a useful way to think about the system? Is it a viable geoengineering solution? What are your views? And you made an interesting comment about you chose not to study this for your PhD.
Yeah, well, I chose not to study it because I didn't even know anything really about it at the time, apart from the limited stuff I'd learned at undergraduate. But I thought, with it all being international waters and nobody having control, who was going to say, yes, this is going to happen? And rather cynically, I just thought, in my lifetime, I don't believe that we're actually going to see this being used. So I decided I didn't want to follow a project on that. That was just a very personal opinion of mine. Because
I think you're probably right.
Yeah. Yeah.
This is never going to be the sole solution.
No, exactly. Yeah.
But it might be a partial solution.
Yeah.
So let's talk about that. About why does it work at all?
So the idea is basically, so it makes lots of phytoplankton bloom because now they're not limited by iron, so they can photosynthesize. So then theoretically, if you have lots of phytoplankton in the surface, once the algal cell dies, they sink out through the ocean to the deep and transport a lot of organic carbon, which has originally come from the atmosphere to the deep ocean. So there's huge sink of atmospheric carbon dioxide. However, this depends on so many things. I mean, there is so much work just going on this carbon export alone, excluding adding iron, and we don't really understand how it's controlled seasonally or annually or in each region, because you have different bacteria will consume the organic carbon and then convert it back into an inorganic form, and then it gets remixed back up to the atmosphere. But it depends on what strains of bacteria, if they're living on the aggregates if they're not. Also zooplankton. Zooplankton consume a lot of these phytochemical aggregates in the operation and then as they sink as well. And the different species of zooplankton have a huge effect on particle flux, because also zooplankton produce fecal pellets when they poo. So as I said, I do a lot of them poo. And these are actually sink quite well as repackaged particles of this phytoplankton originally to the deep ocean. But then zooplankton eat their own poo. So then it can again get respired back into organic, kind of mixed, depending on what's going on with the physics of the ocean. So there are just so many variables that it's just a crazy topic to try and get your head around, and predicting it is really difficult and quite unsuccessful a lot of the time. But what has happened with a lot of the ships, they'll go out for, say, a month on an expedition and they will put a lot of iron overside. However, there's obviously a lag between the phytoplankton blooming and utilising the iron, and then the phytoplankton blooms receding and then dying and sinking out. And often this is not seen by the ships because they've gone home before the sinking flux. Sometimes it just hasn't been observed as much as you would think it was. So, and other times it has been more successful. But there are just so many variables that, as I said, we still don't understand, that it's quite a difficult subject. And yes, as I said, I don't think it's one that's really going to sort everything out for us, unfortunately. And also we don't know what the effects would be. If we did get rid of all of this atmospheric carbon, we don't know what the other side effects that would happen in the mid-waters. Yeah.
Good. John, do you want to comment on that?
Yeah, something that interests me, and next week, week 5, we're going to look at how we explore the ocean and the logistics of carrying out research at sea, which Emma's just touched upon. And I think this iron fertilization work is a really nice example to think about some of the issues involved. So first of all, when you're planning an expedition, you have to have permission. Diplomatic clearance if you're going to someone else's waters, but if you're working in international waters, you've got to make sure that what you're wanting to do doesn't break any international laws. And there are very strict protocols governing dumping of material at sea. And if iron fertilization is categorized as ocean dumping, then the UN can stop you from doing it. And in fact, we have seen the UN actually block research expeditions by some nations to carry out these experiments. So there's a sort of a whole legal area that has to be negotiated. And then the simple logistics of it. So as Emma mentioned, first of all, going out, putting the iron over the side, you've seeded potentially a little patch of ocean, but of course that patch of ocean isn't static. It's moving around with the currents and so on. You've got to be able to track it, to keep up with it, to monitor what's going on in that patch, to see the response, compare it with outside the patch. So logistically, just practically, it's very challenging work to do. And in addition to putting the iron over the side, typically put some sort of tracer over the side that stays at a concentration that you can detect. I mean, the iron might change, you'd expect as part of the experiment. So you need to know you're in the same patch of water where the iron was and it's moving around together. So it's not easy to do technically. And then, as Emma mentioned, the big uncertainty, yes, these experiments have been done and they have seen stimulated blooms of plankton at the surface. But what happens to that carbon that was drawn down ultimately? Does it get exported? Does it actually get taken away from the surface ocean down to the ocean floor? And ultimately, we would hope, if this is a geoengineering solution, locked away for the foreseeable future on a greater time scale than the kind of climate responses that may be involved. Now, just to demonstrate that is, again, technically very difficult. In theory, you could do it using equipment called sediment traps, which are like a gigantic funnel. And what you would have to do is have a very, very long ocean mooring, thousands of meters rising up from the seabed, and at different heights up it, you would put these funnels. Any material sinking, like your zooplankton poo, would end up going down into the trap and being collected and preserved in a pot for us to analyse. How much carbon is there? What's the nature of it? Where has it come from? Has that flux changed over time while this bloom has happened and died, and so on? So you've got to put out these arrays. You'd have to have them confidently in the area that will capture the export of this bloom. When this is all shifting around with the ocean currents and so on at the surface, very, very hard to do, to demonstrate that actual outcome and investigate it. So I think the experiments have shown a response at the surface, but for all these reasons of uncertainty in food chain and recycling and so on, what's the actual impact on the export? And then my interest as a seabed biologist is, what will be the response be if we do see an increased organic flux to the seabed as a result? What will be the changes that we're actually forcing on that system at the seabed and its consequences? There will be consequences and changes potentially. They may be something that you know is no risk to us and much less than the risk we're trying to mitigate against. But there are a whole load of questions that come from this. So it'll be interesting to see where this goes. But I think a lot of the initial optimism, there were even companies out there sort of, you know, you could trade in carbon credits and they dump a certain amount of iron in a bit of the ocean and claim to be drawing that amount of carbon out of the sea, out of the atmosphere. A lot of the initial optimism, I think, has waned as people have started to really get into the complexity of the issue.
Yeah, one of the questions is how do the ecosystem respond if it has no capacity to remove all of this exit carbon?
And that question is just too big for the scope of many research cruises that would just be doing the dumping and the tracing and the export. And then to have you need a whole nother ship with all the people doing the ecology work on it.
So that's why we look at natural systems. So I was involved in a project around, actually it started in the 1990s, around the Crozet Islands in the Southern Ocean.
Yeah.
And so the Crozet Islands are volcanic islands that are volcanic rock is full of iron, naturally. And that rock is very reactive, it weathers very quickly. And so there is a stream of iron coming off these volcanic islands naturally. That stream of iron fertilizes a bloom of plankton annually, which we can see from space. You can see satellite images that have really bright chlorophyll colors for maybe 60 to 100 kilometers wide north of the islands. And so what we did was went and looked at this natural iron seeding event. And so it's been going on for thousands of years. So when we looked at the sediments on the seafloor and the benthic organisms that are living on the seafloor, they've naturally been exposed to this extra carbon dumping because of the natural iron fertilization. That was really interesting. And it allowed us to quantify the difference between the fertilized area and the unfertilized area, the different organisms that live there. The number of organisms that lived underneath the fertilized water were phenomenally higher than the unfertilized normal Southern Ocean. So again, studying natural systems is actually quite a good way of getting at this problem. It still says that yes, fertilizing with iron does increase carbon flux, but it's a complicated system. There are too many risks associated with perturbing that system for the benefits you get for the atmosphere in most people's judgment. So watch the space. It's not going to go away. I think we'll carry on researching this area, but I don't think it's going to be the big solution that perhaps was posited in sort of like the 1990s.
So maybe I made a good decision 2 years ago.
Yeah, I think your question is much more relevant.
And possibly it can work in some areas, but not in other areas.
Yeah. Let's get on to hydrothermal vents, because that's what we've been talking a lot about. Obviously, I'm a chemist and I study these things from one perspective. John was a biologist, he studies them from another perspective. So we're going to try and answer some of the questions that have come up about how many of these things are down there. Now, this started by a discussion, I think that came out of, let's see, I think it was Tim Thornton again. Okay, so Tim pointed out that I made this comment that we can consider these hydrothermal vents to be the water softener of the ocean. They're stripping the magnesium, the sulfate from seawater, and they're sort of, maintaining the composition of seawater that we see. So I made this very bold statement, and I think we can back that up with some detailed studies that we look at, because we know how long it takes the oceans to go through the ocean crust. We know it takes about 40 million years for the entire ocean to go through that water softener system. Tim makes a really good point. How many of these things are down there to be doing this? There must be a lot more than perhaps we've implied from our videos. So over to John here.
Yeah, so it's a very good question. There is an ultimate upper limit on how many there are, because we know something about the rate at which our planet is cooling through the crust. And so the crust is cooling in several different ways. It's cooling through conduction of heat across the crust, but also this sort of convective circulation that the vents are part of. So from geophysics, we can get an upper limit on how many there could be, because there's a finite amount of heat driving all of this. But then that convective cooling that vents are a part of can happen in several different ways. We can have what we call focused venting at these high temperature systems where there's a couple of football pitches in area, the spectacular chimneys, the very, very hot black smoke fluids coming out. And then there's also much more general diffuse flow through the crust, cooling the crust, and slightly different reactions involved and so on, taking place over more widespread areas. So the question is, yes, we can place an upper limit on how much hydrothermal activity there is from the cooling of our planet, but ultimately, how is it partitioned amongst high temperature vent fields and general diffuse flow is still an open question to some extent. So we base that on areas we've explored, how many vent systems have we found, how does that relate to the volcanic activity of that particular section of ridge, and those relationships have been sort of plotted. But I have to say, we undoubtedly are going to be revising those estimates. So for example, the Von Dam vent field that you saw when we looked at the vent fields in the Cayman system, that's a high temperature system, 215 odd degrees where the fluid is coming out. It's not on the center of volcanic rift. It's not giving out the telltale signals that we used to use to find vent systems, and indeed it's not in a location we would have gone looking for vent systems 20 years ago. We wouldn't have thought they were possible, and yet it exists. So there are probably quite a lot more Von Dam type systems potentially out there associated with a particular type of underwater mountain along a lot of the mid-ocean ridge. So how many vent fields there are, we have an upper limit, but we've only seen just more than 200 visually confirmed. We've got signals in the water, another 400 or so that we suspect are out there, where we know roughly where the location is, but we haven't actually got to the seabed to see them and confirm that. But then there's still a lot of exploration to do. So we are operating in that area of uncertainty between, yeah, there's an ultimate upper limit, but exactly how many, can't say yet.
Go on, have a guess. No, I think people do this.
Yeah. You know. So based on our previous experience, estimates I'd seen would put it at about 2,000 worldwide. But then again, I do think that these off-axis systems, which were like the Von Dam, which we didn't expect previously, there could be a lot of those out there. And that's actually important for understanding the chemistry. The chemical flux is actually the chemistry of the Von Dam system is a little bit different, but it's also very important for the biology, because of course at that system we have populations of animals. Now in the case of the Cayman, where we've got the deep ordinary traditional type vent system, we've got the Von Dam, we have one species shared between the two, the shrimp that you've seen, population of both sides of the same species. So if we think about how big are the actual population, what we call the metapopulations of these animals, where they're split amongst, dotted amongst the different vent fields, as we find more sites, obviously there's a bigger metapopulation out there. And oddly enough, when we do population genetic analyses, one of the answers we often get, one of the questions we can ask is how big is the effective population size? How many millions of these types of animals are out there sharing genes from generation to generation? And generally, when we do this, we get vast, vast numbers. And we think, well, we haven't seen enough sites that even approach those population sizes. There must be other sites out there that we don't know about. And my suspicion is it'll be systems like this off-axis that we previously wouldn't have gone looking for, but they are there.
That's really interesting. So I think also the fact that you said 2,000, I heard you, is that it's not 10,000, and it's not, we've seen, as you say, 240 something, haven't we? So we're nowhere near the total number yet, but we've had a good crack at it. Early on in my career, I think we could have counted the number of sites we knew on one hand. So we've come a long way in the last 20 odd years in terms of clocking up more and more known vent sites on the seafloor. But each one tells us something new in terms of species, or in terms of chemistry, or in terms of the geological setting, so we're still at the stage of exploration and actually finding out more about the seafloor. Question leading on from that comes from Heather Evans. And again, it's just one of those biological chemical ones, so you're going to have to help me. Heather says, does seaweed use iron to photosynthesize? Assuming it does. Does the amount of floating seaweed in the Sargasso Sea have any correlation with the iron vented from BB and Von Dam areas? So I think that's a really interesting question.
Right, it is a really good question.
So first of all, does seaweed use iron?
Yes, so iron is really important for photosynthesis. So yes. So the question is, we've got these deep sea vents, they're producing a lot of iron. What impact does that iron have on biological activity in the oceans? Is a really important question. And again, 20 years ago, I think the answer would have been, oh, the iron doesn't go very far. We have the vents and they produce some iron, but it precipitates out into mineral particles and rains back down to the seafloor. And it never goes anywhere that it will really influence. Certainly doesn't get up into the surface ocean. That was always the accepted wisdom. But I think one of the big changes we've seen recently is understanding how iron persists in different forms in the ocean. And it is possible to package iron up with other things and for it to persist much, or to last much, much better, and to be carried much further. And now there's a lot of research that's been going on looking at this wider impact of iron from hydrothermal systems, and potentially in some areas it would have an impact. In the case of the floating weed of the Sargasso Sea, it's, I think, even on our current understanding, a little bit too remote from these cave vents and right up at the surface. But I think there are places where the iron, or we're now appreciating the iron is having a greater impact potentially, and because so much of the ocean as well, we're now understanding is iron depleted, iron limited.
So if we go to the Southern Ocean, for example, in the Scotia Arc, some of the volcanic calderas there have hydrothermal vents in them. So this, in the Kemp caldera, the hydrothermal vents there are probably in only about 500-600 meters depth. And the plume goes up a few hundred meters into the water column. So you can imagine with the very stormy winter mixing you get in the Southern Ocean, that iron that is stabilized in the water column as maybe iron sulfur particles, maybe tiny particles, but still particles, can be mixed up to the surface ocean. Those particles will disaggregate and supply iron to the surface ocean where there is a significant iron limitation. Similarly, in the Bransfield Strait, we've shown that the sediments there are essentially leaking iron from the seafloor, probably via worm conduits. So it's probably the biology that lets the iron out of the seafloor. Again, because it is so stormy in the Southern Ocean, what we've got winter mixing depth is very, very deep. So the wind mixes the upper water column down, if you like, in training and bringing that iron up from the deep to the surface. And we can see these, again, this chlorophyll bloom downstream of the Bransfield Strait, which we think is probably coming from a variety of sources, including these hydrothermal vents. So I think that, again, we're beginning to understand that iron is much more persistent in the ocean than it ever was. Okay, we're running out of time. I'm where it's nearly 1:45, so that's 45 minutes for this Google Hangout. So I think we're going to wrap up now. We're going to see you again in 2 weeks' time. So same time, same place, 2 weeks' time. And the conversation that time is going to be about the material from weeks 5 and week 6. So John, do you want to say what that's going to be?
So coming from week 5, how do we explore the oceans and the logistics involved, and some of the decisions and issues to think about in planning and running field work out on the oceans. And then in week 6, we'll be looking at the future of the oceans and the impact that we have been having for a long time on the oceans. Most of the time, of course, hidden to us, just as the bottom of the ocean has been hidden to us until comparatively recently. And what do our activities mean for the future of the oceans potentially?
Okay, so we welcome you all then, and if you've got any questions you want to put either in the forum or tweet to us, we'll make sure we'll answer those questions in that session, and you'll see us all, or some of us all, in 2 weeks' time. So thank you very much from us, and goodbye.
Bye.
Bye-bye.